Good day, my dear learners. We are still on cell and its environment. We have looked at diffusion and now we will be looking at osmosis. Osmosis is also known as the process of movement of water molecule. The process of osmosis is more or less similar to that of diffusion, but it is more technical. Whereas the process of diffusion involves the movement of gases, liquids, or solids, which could be in the medium of air and liquids. But osmosis involves the movement of water molecules across a semi-permeable membrane. I would love to let you know the meaning of semi-permeable membrane. When we talk about semi-permeable membrane, we mean a membrane that allows certain materials to pass through and does not allow others to pass through. In the body, there are many semi-permeable membranes. In fact, in general, biological membranes are semi-permeable. That is, they allow certain materials such as water to pass through and they don't allow others to pass through. Now, we'll be looking at the definition of osmosis. During the course of this lecture, we'll be having several definitions, so I want you to pay attention carefully. How can we define osmosis? Osmosis can be defined as the diffusion of water molecules across a semi-permeable membrane. It can also be defined as the movement of water molecules from a region of low concentration of solute to a region of high concentration of solute through a semi-permeable membrane, separating the two solutions until equilibrium is attained. The solution which loses water in osmosis is said to be hypotonic, and the solution that gains water is said to be hypertonic. But when a state of equilibrium is established, the solution is said to be isotonic. By isotonic, we mean that there is no net gain nor loss of water. I will be using a diagram to show you the process of osmosis. I will show you the movement of water molecules across a semi-permeable membrane. So now this is our diagram. From the above diagram, you can see that solution A has a very high concentration of solute dissolved in it, and the solutes are being shown to you. Those are the big solute molecule of salt. You see, they are in high concentration in solution A. Now, moving to solution B. Solution B has smaller amount of solutes dissolved in it. The solutes in solution B are also what outlined to you. Since solution A has a larger amount of solute, it is known as the concentration what solution, while solution B is known as the dilute solution. The solution B contains a high concentration of water molecules. So, therefore, solution A with higher concentration of solute has a smaller amount of water left for dissolution of more solute, while the solution B with lower concentration of solute has more water available. What this means is that the solution B will have a higher concentration of water than solution A. And since these two solutions are separated by a semi-permeable membrane, water will move from solution B into solution A through the semi-permeable membrane. You can see the arrow at the bottom there that is showing the net movement of water molecules. That arrow shows you that water is moving from solution B to solution A. It is important for you to also note that any given solution has a certain amount of osmotic pressure. What is osmotic pressure? Osmotic pressure is the force that draws water into a cell. The osmotic pressure of distilled water is equals to 0. Also note that the higher the amount of solute dissolved in a solution, the higher the osmotic pressure of that solution. And from the diagram that you can see, the osmotic pressure of solution A will be higher than the osmotic pressure of solution B. And this is because there is greater amount of solute present in solution A as compared to solution B. Osmotic pressure can also be defined as the pressure required to stop water from diffusing through a membrane by osmosis. Now, this will bring us to another definition of osmosis. Osmosis, by the virtue of the osmotic pressure, can therefore be defined as the movement of water molecules from a region of lower osmotic pressure to a region of higher osmotic pressure through a semi-permeable membrane. Referring back to our diagram, as water is being drawn into solution A from solution B, the osmotic pressure of A is slowly being reduced. A time will come when the osmotic pressure of solution A will be equals to that of solution B. And at this time, the two solutions will be said to have reached an osmotic equilibrium. And when these two solutions are in equilibrium, the rate of movement of water from A to B equals the rate of movement from B to A. I will be showing you a diagram to illustrate what I have just said thus far. This diagram is a modification of our previous diagram. We see that solution A is now in equilibrium with solution B. There have been net movement of water from solution B to solution A. We can see now that the number of solutes are actually equal on both sides. That is, the number of solutes in solution A equals the number of solutes of what? Solution B. And thus, the rate of the movement of water from A to B equals the rate of movement from B to A. Having shown you the processes involved in osmosis, I will be telling you the importance of osmosis in living organisms. Osmosis is very important in living organisms in the following way. Number one, in plants. Plants absorb water from the soil into the root hair by osmosis. Plants absorb water from the soil into the root hair cells by osmosis. 2. Water from one cell into another in plant and animal moves by osmosis. That is, the movement of water from one cell into another cell in plant and animal is by the process of osmosis. 3. When the stomata opens and closes, that is, the opening and closing of stomata takes place when guard cells take up or lose water by osmosis. 4. The turgidity that is being experienced in plants and animals is through the process of osmosis. 5. The absorption of water into the colon of animals occurs by osmosis. 6. The reabsorption of water in the kidney tubules of mammals is by osmosis. During the process of excretion, there is the absorption of water in the kidney tubules of mammals, and this process is by what? Is by osmosis. So we have looked at the importance of osmosis in living organisms. It is important for us to mention the differences between diffusion and osmosis so as not to confuse the concepts together. So we will draw a table. On the left side we have diffusion, on the right side we have osmosis. 1. The process of diffusion involves the movement of molecules of gases, liquids, and solids. But the process of osmosis involves the movement of molecules of water only. 2. In diffusion, movement of molecules need not occur through a semi-permeable membrane. That is, a semi-permeable membrane is not involved. But in osmosis, the water molecules always passes through a semi-permeable membrane. Number 3. In diffusion, molecules will diffuse into any space accessible to them. But in osmosis, water molecules move between two solutions of different osmotic pressure, and this will lead to the establishment of an osmotic equilibrium. Number 4. Diffusion occurs in both living and non-living things. But osmosis occurs only in living things. let us describe theoretically an experiment on osmosis. This experiment will be also shown in a live practical session, but now for the purpose of understanding of this concept, we will be describing it theoretically. And it has been shown over time that questions like this comes out in exams, or when you are drawing your diagram in exams, you shouldn't shade. There should be no shading. Also, your lines are to be thin lines. And when you are labeling, the labeling should not cross each other. Now to our experiment on osmosis. To perform any experiment, you need to know your aim, and you need to have some specific materials. What is the aim of this experiment? This experiment is to demonstrate osmosis using yam tuber as living plant material. And what are the materials we will be using? The materials include two thick slices. The materials include 2 thick slices of yam, strong salt solution, distilled water, and a trowel. I would drop a diagram to show and illustrate the methods that will be used in this experiment. Now, our 2 thick slices of yam tuber A and B. When we have obtained them, we peel off their skin and make a large cavity in the middle of each. So we can see the cavity in both yam tubers A and B. Now, a strong solution of salt is kept in yam tuber A. That is the strong solution of salt in yam tuber A. And the salt solution is marked T1. It's marked T1, while yam tuber B is left without salt solution. And these serve as the control experiment. So we can see yam tuber B, the cavity there is empty without salt solution. Now, they are both kept in a trowel, and this trowel contains distilled water. That is the distilled water that is shown in the diagram. And this experimental setup is left to stand for some hours, and our results will be obtained. So after some hours, the result that we will observe is that the salt solution in tuber A has risen to level T2, while no water enters the yam tuber B. In conclusion, the salt solution in yam tuber A has a higher osmotic concentration than the water in the trough, and exerted a higher osmotic pressure on the water. The water with lower osmotic concentration moves through the cells of the yam, which acts as a semi-permeable membrane to the cavity of the yam. Hence, there is a rise of the salt solution in yam tuber A. But in yam tuber B, water could not be drawn into its cavity because it contains no salt solution, and the cells of the yam have lower osmotic concentration and osmotic pressure than the water in the trough. This brings us to the end of our discussion in osmosis. Right about now, we will be taking some questions on this concept in order to aid our understanding. Our first question: which of the following statements is not true of osmotic process? A. There must be a selectively permeable membrane. B. The two solutions must be of different concentrations initially. C. It involves only the movement of water molecules. D. Equilibrium is reached when there is equal distribution of water molecules. E. The two solutions are of equal concentrations at the beginning of the experiment. Our answer to this question is simply E, which says the two solutions are of equal concentration at the beginning of the experiment. Why is this our answer? This is because at the beginning of osmotic processes, the two solutions are not of equal concentration, but rather they must be of different concentration. For osmosis to occur, two solutions must be of different concentration, and these two solutions must be separated by a semi-permeable membrane. So our next question: osmosis can be defined as diffusion of A. Atoms and molecules through a membrane to an area of higher concentration. B. Water molecules from a dilute solution to a concentrated solution across a permeable membrane. C. Water molecules from a dilute solution to a concentrated solution through a semi-permeable membrane. D. Perspiration and excretion. Our answer is C. Water molecules from a dilute solution to a concentrated solution through a semi-permeable membrane. The reason for this is that osmosis is actually the diffusion of water molecules from a dilute solution, that is, a solution with less solute concentration, to a concentrated solution, that is, a solution with more solute concentration. And these processes happen through a semi-permeable membrane. So our next question: in which of the following yam tissues will osmosis occur? In which of the following yam tissues will osmosis occur? A. Boiled and peeled. B. Raw and peeled. C. Boiled and unpeeled. D. Roasted and peeled. Our answer: from the experiments we discussed during the course of this teaching, we saw in the method that we will use a raw and peeled yam because a raw and peeled yam can act as a semi-permeable membrane. So this means our answer is B, raw and peeled. So our next question, I will show you a diagram, and with this diagram you will answer the question that follows. Now the first question, it says after an hour the level of water in the tissue funnel will A, rise, B, fall, C, remain the same, D, double. This diagram is simply illustrating the process of osmosis, and osmosis is the movement of solvent, that is water, from a region of less solute concentration to a region of more solute concentration through a semi-permeable membrane. In the experiment you can see the distilled water, which is the less concentrated solute solution, moves through the semi-permeable membrane into the sugar solution, which is the more concentrated solute solution, and as a result of this, the water level of the sugar solution, which is in the tissue funnel, will go up. That is, it will rise. This brings our answer to A, rise. So our next question, it says the experiment above is used to demonstrate the process of A, transportation, B, water culture, C, diffusion, D, osmosis. The answer is osmosis. And if you have listened to the first explanation, I'm sure that you will know the reason why. We have come to the end of our examples. I will be dropping some exercise for you to try your hands on. This exercise we gotten from our textbook on hidden facts in biology. You can see the questions there. You can try your hands on them. The answers to these questions are at the back of the book.